Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. Now we are going to proceed towards the microcirculation. As you all know that we have already studied the arterial system and the venous system. Now the microcirculation is very important because the capillaries are the part of the circulation which deal with the delivery of nutrients as well as taking up of the waste products from the tissues. And whenever the capillaries they are supplying something or taking, the pressures are very important, because when there is increased pressure in the capillaries as compared to the tissues, the capillaries they will filter out, otherwise they will absorb. So in this chapter we are going to have a look upon the capillaries, their functions, their features, specialized feature of certain capillaries in various areas of the bodies, like kidneys, like brain, like intestines. And then after that we are going to study the pressures in the capillary. So the microcirculation, it involves the smallest arterioles, the metarterioles, which are encapsulated by sphincters, which are known as the pre-capillary sphincters. Then we have the capillaries. There are the capillaries have a venous end and an arterial end. At the arterial end, the capillaries they supply the oxygenated blood and nutrients, and at the venous end they collect the waste products and carbon dioxide from the tissues. Then there are small venules. Now this is the structure of the capillary system. As you all know from the study of histology, that arteries they have three layers. The internalmost layer lines the lumen, the tunica intima, and the tunica intima, the blood leaves the tunica intima to the smaller arterioles and the meta-arterioles, and from the meta-arterioles it enters the arterial end of the capillary system. In the capillaries are different from the veins and arteries in that they are lined by a vascular epithelium which is single-celled, unicellular vascular epithelium. At this point, the arterial end, you see that the color of the blood is red and it is oxygenated blood, whereas as far as it is moving down the tissues towards the venous end, it is becoming blue. So it is carrying deoxygenated blood now. With the waste products of the deoxygenated blood, it carries to the venous system. Now the structure of the microcirculation and capillary system involves the arterioles, the meta-arterioles, and the pre-capillary sphincter, as we have already discussed. The capillaries, as far as the cross-sectional area is concerned, they have the widest cross-sectional area, that is about 2500 cubic centimeter. Now despite the fact that the aorta is the largest vessel, its cross-sectional area is only 2.5 centimeter square, whereas that of all the capillaries is 2500. So it means about 100 times more, or rather 1000 times more as compared to the aorta. Now this diagram is from some other book, not from Guyton. So I have moved that diagram away. We will study that later. Now in this particular section, as you can see that the arteries and the veins and the capillaries have the widest cross-sectional area. They have, and this wide cross-sectional area, you all know, is necessary for sufficient and efficient supply of nutrients to the tissues and collection of waste products from the tissues. Now this is the diagram. The capillaries which arise from the meta-arterioles, they are controlled by pre-capillary sphincters. These pre-capillary sphincters control the pressures of the blood which is entering the capillaries, the capillary system. Now here is another diagram of these meta-arterioles. These pre-capillary sphincters, they gauge, they very precisely monitor the blood flow to the tissues, whether it is increased, whether the demand has increased, so the blood flow increases, whether the demand has reduced, so the blood flow it reduces. Again, this is the same diagram showing the importance of the pre-capillary sphincters, and this pre-capillary sphincter is very important because sometimes when you are exercising, your muscles need increased blood supply, increased oxygen. So the capillaries to the muscle, they vasodilate. So this is the role of these precapillary sphincters, which cause this vasodilation. They just relax. On the other hand, when vasoconstriction is needed, like in exercise, the blood supply to the intestine is reduced. So the precapillary sphincter of the intestinal capillaries, they start contracting, causing vasoconstriction. Now this is a 3D view. This diagram is very important. This is not present in Guyton. I wish you make a 3D view of this concept, that how the capillaries, veins and the blood systems, they are working. This is the arteriole, this is the capillary tree, and this is the vein. So the blood flows from the arteries to the capillaries, and from the capillaries the nutrients, they are supplied to the tissues, and from the tissues it is taken away by the lymphatic system, that we will study later in this chapter. On the other hand, at the venous end, it is not shown here, but the nutrients, rather the waste products, they are collected by the capillaries and then drained into the venous system, which takes it back to the heart. The capillaries, they are the smallest blood vessels. The diameter ranges from 8 to 10 microns. The red blood cells pass through the capillaries in a single file, as I will show you later, that there is no stacking of red blood cells, only they can pass through in a linear fashion. And there are specific types of capillaries in lungs, small intestines. In the lungs, they carry the oxygen and carbon dioxide. The small intestine, they receive the digested nutrients. In the endocrine glands, that they pick up the hormones, and in the kidneys, they cause the removal of nitrogenous wastes. So this is the wall of the capillary. The wall of the capillary has an endothelial cell, a basement membrane which is present here, and then there are small vesicular channels. The presence of these vesicular channels is still contentious, whether they are present or not, it is debatable. However, there are pores which are known as plasma lemmal vesicles and intracellular clefts. These pores allow the nutrient movement between the capillaries, through the capillaries, and this is very important that these pores are present, because otherwise the movement of the nutrients and waste products and oxygen and carbon dioxide will not be very easily possible. Now these pores are specialized in certain organs: liver, brain, muscle, GIT and kidney. Just remember this here, then we will study these in detail after a while. And then another system of circulation known as lymphatic circulation is also present, which collects certain parts of the circulation, and this is drained into the lymphatic system. You have already studied that in anatomy. In this diagram, you can see that RBCs, they are moving in the capillary in a single file. You can't see two RBCs like in a narrow road. When the cars are passing, they can't overtake each other. So the RBCs, they can't overtake each other because of the narrowness of the capillaries. And why this is important? You should guess it. Because these RBCs, each of them is exposed to the capillary wall at every instance. At every instance of time, each RBC is exposed to the capillary wall. Something about the nature of the pre-capillary sphincter: they regulate the flow of the blood to the tissues, tendons and ligaments. They are poorly vascularized, and in the epithelium and cartilage, the capillaries, they are avascular. The pre-capillary sphincter regulate the blood flow to the tissues. So the capillary beds, you have to remember that they are different in each part of the circulation, and they are specialized in different parts. Whereas, just to emphasize, because chapter number 17 is about the control of local blood flow, which we will study in a couple of days inshallah. So these pre-capillary sphincters are very important in managing the direction, the amount of blood flow to some local tissue. Again a repetition. Now the capillary permeability, the endothelial cells, they are held together by tight junctions and desmosomes. As we studied in an earlier diagram, there were intracellular clefts, which were the gaps in the unjoined membranes. Small molecules can enter and exit. Then there are two types of capillary. Continuous type of capillaries, they are most common. Then there are fenestrated, which are being supplied with a number of pores or oval type of pores, oval shaped windows, as I will show it to you in the next diagram. That some capillaries, that most capillaries, they are continuous without pores, whereas some have fenestry. Fenestry means oval windows, small windows, which are for the transport of nutrients. So for example, in this diagram we can see that the capillaries, they are grouped according to their leakiness. Most common are the continuous capillaries. I am enlarging this diagram for you to understand better. So these are the continuous capillaries which have a basement membrane, and these are present in most parts of the circulation. So capillaries which have pores, let me move it upside, they are known as fenestrated capillaries. And here you can see that open fenestra or open small oval windows, they are present for the circulation, and they are found in intestine, kidney glands and choroid plexus. Now there are sinusoidal discontinuous capillaries. You see that this is broken, the cells are not attached properly to each other. There are big junctions within the cells of the endothelium, so they allow high leakiness. Continuous capillaries allow moderate leakiness, fenestrated capillaries more leakiness, and the maximum leakiness is present in the sinusoidal. So as expected, they are present in the liver because the liver has to detoxify and manage a lot of metabolites. So this is another comparison that you can see here. You can very well establish that the continuous capillaries are attached, the cells of the continuous capillaries, the endothelial cells, they are attached to each other. Then there are fenestry, and then there are discontinuous capillaries or sinusoidal capillaries. Now this is a rather magnified structure of capillaries showing the cross section. There are pericytes and the red blood cell. This is just present in single file. Again repeating this point, in the endothelial cells there are present pinocytic vesicles, there is a basement membrane, tight junctions, intercellular cleft. All this you have studied before. So in another diagram you can see that the endothelial nucleus is also present, the endothelial cells are there. Then there are fenestrations here. This is the structure of a fenestrated capillary. So these fenestry or the oval windows, these are present here for easy transport of the nutrients. Now you can see that these capillaries, again repeating the same diagram, they are continuously in touch with the tissues, and this is the main function of the capillaries, that they have to manage the transport of the substances. Now the pressures are very important. After the size of the holes from which the transport mechanism takes place, we will be studying about pressures. So keep this picture in mind. So the permeability of water is taken as standard 1. As the molecular size increases, like that of sodium chloride, the permeability it reduces, and as it keeps on increasing and increasing, the size of the molecules it increases, the permeability it reduces. So when we reach the hemoglobin, the permeability is only 1/100th to that of water, means 0.01, and albumin, which is 69,000, the permeability is reduced to 1/1000, 0.001. Now why this difference here? This is because of the charge on albumin, that it is negatively charged, so it is not very easily permeable through the capillary pores. Now the flow of the blood in capillaries is known as vasomotion, which is waxing and waning of movement, increase or decrease of movement. It is very well calculated, and it is managed by the oxygen concentration. When the oxygen concentration is here, the overall function is averaged. Now there are four routes into and out of capillaries. So when we are talking about the routes of capillary permeability, there are four routes into and out of capillaries. One is direct diffusion, then other route is through the intercellular cleft, then through cytoplasmic vesicles, and finally through the specialized pores known as fenestrations. Then we are talking about low permeability capillaries, which are present in the blood-brain barrier. They have very tight junctions and there are no intercellular clefts. The vital molecules, they pass through highly selective transport mechanisms, and it is not a barrier. This blood-brain barrier is a barrier against most of the molecules except oxygen, carbon dioxide, and some anesthetic agents. The sinusoids, which we studied about a while ago, they are present in the liver. They are wide leaky capillaries found in some organs. Usually fenestrated, intercellular clefts are wide open, and they are also present in bone marrow and spleen. They have a larger diameter and a twisted course. Now we are talking about the sinusoids. They have leaky channels. Some of them are present within the endothelium, and there are some tight junctions as well, but most of the times they are leaky. Now this is an important diagram that you will study in more detail in second year, that what is the amount of, let me enlarge it, different types of fluid. So there are intracellular fluids and there are extracellular fluids. In the intracellular fluid there are 25 liters. With intracellular, the total body water, you know that in a 70 kg man, 2/3 of that is water. So out of 70 kgs, 42 kgs or 42 liters are water. Out of those 42 liters, 25 liter is about 25 liter is intracellular. The rest is extracellular. Out of the extracellular fluid, 3 liter is blood oblique plasma, and 13 liters is intercellular fluid, interstitial fluid, and there is transcellular fluid. So 2/3 is intracellular and 1/3 is extracellular. The intracellular fluid is about 25 to 28 liters. The total body water is 42 liters. So this is all for this lecture. The second portion of this lecture will be uploaded later, and then we will study about the hydrostatic pressures. Thank you very much.